Welcome to this short presentation introducing the Aridia real-time PCR tests, CTK Biotech's brand new line of molecular diagnostic products. Our first wave of tests target tropical diseases and provide accurate and sensitive detection of virus during the early stages of infection as an aid in the diagnosis of these diseases. CTK Biotech's current IVD offerings have long included an extensive range of rapid test and ELISA kits for the screening and detection of infection, as well as rapid test external controls to monitor test quality. The addition of the Aridia real-time PCR line expands this product range even further and allows CTK Biotech to offer a complete line of diagnostic solutions for every stage of infection. PCR testing is needed because it offers the earliest confirmatory testing method that detects the pathogen before it's cleared by the immune system during an infection. It also offers the added benefits of increased sensitivity compared to serological tests, as well as specific detection with no cross-reactivity to other pathogens. The first wave of Aridia real-time PCR tests are designed for the detection of Zika, dengue, and/or chikungunya viruses, which are transmitted by the same mosquito vectors and can cause similar clinical symptoms. Detection of these viruses using the Aridia real-time PCR test confirms an infection, and you can even test for all three viruses at the same time using the Zika, dengue, chick multiplex test. The kit comes with 8 well PCR strips with the reaction mix stabilized within each well, resuspension solution to resuspend the reaction mix prior to use, and optical caps to cover each well during the PCR run. Also provided are positive and negative controls, which are crucial for interpretation of PCR results and must be included with each run. Finally, PCR-grade water is included to rehydrate the lyophilized positive control. The procedure to run the Aridia real-time PCR test is simple, user-friendly, and fast, requiring only 4 easy steps. Prior to using the kit, all specimens must be extracted using a nucleic acid extraction kit. In the first step, simply resuspend the stabilized PCR reaction mix in each well using the provided resuspension solution. In step 2, add 5 microliters of your extracted specimen or positive or negative control. Third, load the wells into your thermocycler and run the PCR program. Finally, interpret the results after the run is completed. That's all there is to it. Aridia real-time PCR tests provide unparalleled stability, integrity, and convenience. Because the positive control and the PCR reaction mix in each well are lyophilized, the kits are stable at room temperature for up to 24 months. This shelf life is longer than most competitor kits, which require frozen shipment and storage and generally last 12 months. The stabilized format of Aridia kits means worry-free transportation with no concerns about the shipping time, the weather, or product performance upon arrival. The room temperature storage saves valuable freezer and fridge space for other important needs and provides peace of mind from power outages or freeze-thaw cycles. Other kits require calculating the volumes of reaction components to add, pipetting them into a master mix, and dispensing the reactions into individual tubes. With the Aridia real-time PCR tests, the PCR reaction components are pre-mixed and ready to use in each well, so there is less pipetting, fewer errors, less time spent, and reduced potential for contamination. The Aridia real-time PCR tests run using the same thermocycler program with results in about 1.5 hours. This feature allows different tests such as for Zika virus or dengue virus to be included in the same PCR run, so there's no waiting for one test to finish before running another. Aridia test kits are designed to fit seamlessly within the existing equipment infrastructure in each molecular diagnostics laboratory. The tests are offered in high or low profile tubes, providing compatibility with many real-time PCR thermocyclers commonly found in molecular diagnostic laboratories, which are listed in each product insert. The Aridia Zika Dengue Chik Real-Time Multiplex test is designed for the simultaneous detection of all 3 viruses in the same test and is particularly useful for patients experiencing non-specific clinical symptoms in areas where these pathogens are endemic. The efficient diagnosis of these viral infections without the need to run multiple tests can help doctors make informed patient management decisions faster. In various studies with clinical specimens, all Aridia real-time PCR tests demonstrated 100% overall agreement with commercial and/or in-house reference assays, as well as no cross-reaction to common arboviruses such as West Nile or yellow fever. The ability of these tests to detect their respective targets was also confirmed with several different viral strains. The Aridia Zika virus real-time PCR test was also subjected to 2 external evaluation studies in Brazil and Colombia. The test demonstrated 100% relative sensitivity and specificity in both studies and featured a higher sensitivity and specificity over the competitor kit or in-house reference assays. The Aridia real-time PCR test positions CTK Biotech as a one-stop shop for all your diagnostic product needs. From confirmatory detection of pathogens early in the infection with the Aridia kits, to screening and detection of antibody response later in the infection using on-site rapid test and recombinase ELISA kits, CTK Biotech offers a full complement of diagnostic solutions. CTK Biotech has diagnostic products that fit every type of facility and budget, from large central laboratories with extensive resources and equipment to small laboratories with limited facilities. CTK Biotech products are backed by extensive technical support channels and resources that provide product assistance, knowledge, and troubleshooting, and ensure customer satisfaction. Technical support can be reached by email at tech@ctkbiotech.com or by phone, and resources are available on the CTK website. Thank you for your attention.